Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. This is Dr. Habib, and I want to welcome you back to the Health Upgrade Podcast. Today's guest is Ryan Smith. Now, Ryan is the founder of TruDiagnostic, and I can tell you that this conversation was quite in-depth and quite empowering. I think from a very clinical perspective, as a clinician, I had a really amazing time listening to Ryan speak about how we're actually able to use DNA and to understand how we can change the expression of our DNA using epigenetics. And that's what TruDiagnostic does. TruDiagnostic is a cutting-edge company that offers personalized and preventative healthcare products and services through leveraging advanced technologies such as AI and genomics. And they started in 2020, and since then, they've created the largest private epigenetic database in the world with over 70,000 patients. They've published over 30 academic papers, been awarded 10 plus patents. I can also say very clearly after this conversation that there's a lot of amazing, phenomenal information coming out with regards to epigenetics and how we can help express our genetics in a more healthful wellness fashion manner and really use it to upgrade our health. They have some amazing offerings, which we talk about at the end of this episode. And I'm really, really excited to share this episode with you because this was such an engaging conversation. I think there's some major insights and some real opportunities coming out for us to empower ourselves and to take on this idea of upgraded health. So without further ado, here we go. Ryan Smith and TruDiagnostic. Ryan, it's wonderful to have you here. Thanks for joining us.
Yeah, thanks for having me. Excited to talk all things DNA methylation.
For a lot of people out there, they may not fully understand what DNA methylation is. And I think a good starting point for our conversation can just simply be to explain what is DNA methylation in its most basic definition.
Yeah, definitely. And this is certainly a topic that has a lot of confusion. I think a lot of people are familiar with methylation and DNA, things like, do I have a high homocysteine or do I have certain variants which might prevent this biochemical process? But really what I'm talking about when I mention DNA methylation is sort of the larger field of epigenetics. Sort of epi meaning above the genome. These are changes which happen to our gene expression. And I think the best way to explain it is by sort of talking about our cells and our tissues, right? Our brain and our skin and our heart all have the same DNA sequence, but they behave very differently. And the way they behave very differently are what genes are actually turned on and which are turned off. And so that's sort of, I would say, the broader class of epigenetics is how we, take that hard-coded information and then interpret it to producing signals. Really DNA methylation is sort of that off switch. We sort of consider it, whenever it's generally attached to certain gene regions, you transcribe that gene less. And so it becomes less active. And so I think that this larger field of epigenetics is certainly having a revolution right now in terms of being able to read patterns. And those patterns can be super relevant to really everything in health.
I think patterns are a really important thing to understand. And JP and I have this interesting kind of definition and how we talk a little bit about how the DNA is used by different cells in different organs and different areas. And this whole concept comes down to the DNA is the same in every single cell. We have the same owner's manual / blueprint in every single cell. But liver cells, hepatocytes, are going to do something very different than alveolar macrophages, are going to do something very different from neurons, are going to do something very different from an epithelial cell. And so the concept that we've created is those patterns that we want to have functioning really well, that we want to have turned on in certain cell types. Methylation is a tool that they use to essentially bookmark a page in that manual or bookmark an area in that blueprint. Is that what you would say kind of fits that definition?
Yeah, I certainly like that analogy. I think that one other that works really well is the hardware versus software, right? Where DNA is that hardware, but the software is different in every cell because, you might want one cell to run the software of a heart cell, while others an immune cell. And so I think that hardware-software analogy is a really good one and explains, I think, some of those differences.
What got you interested in this concept of DNA methylation in the first place, and to kind of dig your career into this trajectory?
Yeah, I've had a very strange career in a lot of different areas, but really after doing some medical school and finishing my degree in biochemistry, I really wanted to do something a little bit more entrepreneurial. So I created a compounding pharmacy that specialized in peptides and proteins, and we grew really, really rapidly. We were the fourth fastest growing company in healthcare because we were really hitting a very niche target of preventative medicine physicians who were really focused on age as a primary outcome. So age has always been something of a focus in the process of good aging. And for those of you who maybe haven't heard of this in a medical context, aging might become across as sort of like a vanity thing, but I think that we consider age, especially here at TruDiagnostic, to be the biggest risk factor for every chronic disease. We think that healthy aging is incredibly important to longer lifespans, but also better health spans, living longer without disease. And so this has been a focus of mine, mainly because I believe in preventative medicine. I believe it's so much easier to prevent, a disease than to treat it. And aging being the biggest risk factor is really the thing that took up a lot of my attention. And so we were doing a lot of really innovative products within this pharmacy to treat this as a criteria. But at the end of the day, we didn't have a great way to measure or predict some of the impact that some of these medications were having. And so the pharmacy we started in 2016 was a few years after even the first of the DNA methylation-based clocks. And so I was always paying attention to these. But as they started to advance a little bit further, I got really excited about the technology in aging, but also about how that technology, I think, could have impacts in other types of healthcare. So we ended up selling our business in 2020, really with the idea of creating TruDiagnostic to specialize as a lab, first off to run these specimens and samples, but then also the most exciting part is to read those patterns. I sort of talk about creating the Rosetta Stone of understanding that DNA methylation landscape. We're trying to find the patterns, and now we can actually do that with artificial intelligence and these machine learning models that weren't even available at the start of our company. And we're making I would say massive leaps and bounds to try and understand what those patterns are. So that's sort of, I would say, my history in it, and it really starts with biological age.
So who is the first person to imagine that DNA methylation could predict aging, and how did that technology sort of evolve?
It really started, I would say, right around 2009. But the person who's credited most with this is Dr. Steve Horvath from UCLA. Steve Horvath was actually a relatively notable figure already in this field of bioinformatics before he even went into the DNA methylation field. But he published a clock in 2013, which is still probably the most widely published clock, which was a multi-tissue clock. So it could take cells from any sort of part of the body and tell you the chronological age of the participant with fairly reasonable accuracy. And this was a bit of a revolution, I think, at the time. Everyone was incredibly excited that our gene patterns of expression were changing in such a precise way that we could actually predict the chronological age of a participant across multiple different cell types. It almost brought up this idea that there's a universally conserved signature for aging.
So that's really interesting, because one of the questions that I have and still have, and maybe you can clarify it for me, is, people's tissue, for example, your kidneys, if you've, gone through, as I have, multiple kidney stones, people who might have high blood pressure, that's not me, but some people who have high blood pressure or other tissue-specific issues might have a different DNA methylation pattern. Clearly, they're going to have a different DNA methylation pattern simply because they produce different proteins. But how is it possible to choose, just for example, blood cells, lymphocytes or something, and be able to extrapolate that all of the cells in the body have reached that specific age? How do you not get confused that from the blood sample, you predict a person's age, but their brain isn't there, their skin isn't there, their heart muscle isn't there? How do you discern that, or how is that?
Great question with, I'm afraid, a relatively long-winded answer. And I think that to address some of the fundamental concerns there, you're absolutely right. One of the worst things about DNA methylation is also the best thing, which is that it is incredibly complicated and changes with everything in our lifestyle, whether that's diet, nutrition, exercise, pharmaceutical ingredients, whether it's how much UV exposure we're getting, right? All of these things change, and so that makes it both good and bad. It makes it good because we can now, if we figure out what those patterns are changing, we can actually figure out exposures. We can figure out all of these things from one data set. We can figure out current disease status, current disease risk. But the bad thing is to get there, to get where it's usable is very difficult because we have to control for just about everything. And anyone who's done any type of scientific research knows just how difficult that is. So there have been a couple different approaches here that I think are important. The first ever clocks are clocks that really we don't use anymore. And I think that this is important to mention because they were trained to predict the chronological age of a participant. So sort of the ground truth that Dr. Horvath was using in those original clocks was the age of the participant. That's how he was training these algorithms to pick their signals. And this was really good if you're trying to predict the age of a participant, which originally it was. It was used at crime scene investigations or dating refugees to see if they were adults or minors. But ultimately, it's not really what we want to measure if we're trying to measure the biological process. So what we call the second generation clocks started to be trained on biological phenotypes. So these are how can we pick up the actual biology and sort of answer the question of, we all know people in their 50s who might look 70 and vice versa, someone in their 70s who might look 50. This is really the process we want to measure, right? It's really what we want to predict because that is what's leading to disease outcomes. So these second generation clocks started to be trained, and their ground truth was really things like blood-based biomarkers we know change with time, or so on and so forth. And these clocks got a lot better. And when I say better, that means they were more predictive of outcomes, meaning that if you're accelerated, you're more at risk, and if you're decelerated, you're more protected. And ultimately, that's really what we want to see. So to take it, I think, JP, to your end question, the blood is generally pretty compatible with most tissues, which is great because it means that hopefully with blood we can. Most tissues, as you mentioned, the brain, not much. There's a 20% correlation between blood methylation and brain methylation, which makes that very difficult, right? So the other thing is that as these clocks have developed, one of the things we've learned is that for biological age, it's so complex, right? There's so many things that happen with aging. The more information we feed into the models, the better our prediction. So that includes diet, lifestyle, metabolites, proteins, all the genomics, whatever we can include in there makes our prediction better. But the other thing is we don't just want to make it our prediction better. We also want to make it more interpretable. Right? We want to make it more usable with that information. And so there are certainly techniques that have been done to do that too. So for instance, now we're just launching a clock actually tomorrow with Yale, which is an organ systems age clock. So we can actually tell you the age of 11 different organ systems, your kidneys, your liver, your heart, your brain. And we have done that by training each organ based on blood-based biomarkers. So we might have 10 biomarkers that we choose to measure your heart health. And we do that in a couple thousand participants across time, create a score, and then train DNA methylation to predict that score. And so what we can do now is actually break down those signals into an organ-specific score based on the other associations. And what we see in our validation when we test these out, we see it works pretty well. People who have high, musculoskeletal aging have worse musculoskeletal outcomes. Same with lung or brain. And that's really where we're getting it to not just more predictive, but more usable as well.
Do people have, you mentioned that the methylation patterns shift pretty dramatically depending on what you're eating, what time of day it is, whether or not you've been exercising, a host of different things. I'm assuming that those shifts are relatively coordinated. That means that if you've been sedentary and you begin running and you start getting yourself in better shape, you're going to see a predictable shift in methylation pattern. And that's really important because even though things are changing, doesn't mean that they don't change in a predictable way that can be used to generate additional information. So I think that's probably a pretty important point. But help me understand how, as an individual, I might be able to use this information to inform me as to how to change things about my life or my lifestyle.
And I think that's certainly a good point. And that actually even brings up what I would call the third generation clocks, where they actually track individuals longitudinally based on their change. So we got the first chronological age, second biological, third biological but longitudinal, which is how we really specialize in getting those signals to make, because again, as you mentioned, they're based on biology, they're predictable, they happen in a coordinated fashion, and longitudinally in a single individual is the best way to find that pattern. And combining that across multiple individuals is the best way to find that pattern. But once we have the tool created, which is no small undertaking, and one that is going to continue to advance, and across all fields, not just aging, which I think hopefully we'll have some time to talk about. But particularly for aging, I think that the idea here is that we really want to quantify your biggest risk factors, right? We always say you can't manage what you can't measure. And we've determined that these tools are very good through validation. So we take the tools we've developed, we take a completely different group and cohort, and we show that our assumptions are true, which is our assumptions are that faster aging leads to increased risk, and slower aging by this metric leads to decreased risk. So ultimately, we want to figure out with you how we can get you lower, right? How can we decrease your aging? And there, I would say, previously with some of these clocks, it was really hard to make any personalized recommendations. This is something that is still probably just within the last 6 to 8 months that we've been able to do, because we can tell you, for instance, if you're older or younger. But unfortunately, a lot of the recommendations we know for aging are the exact same for everyone, right? They're the things that we already know, generally improve. More exercise, right? Better diet, nutrition, less processed foods, caloric restriction. These things work, generally across everyone. And that was helpful to sort of see where you're at and to maybe even gauge where you're going. So you could, for instance, try an intervention and then see, did that help or did that hurt? But ultimately, we really want to start to give you recommendations on the outset, the first time you take that test. And so now we've been able to do that by, again, breaking down the signals a little bit further to say that this signal is driving your aging or that signal is driving your aging. So, for instance, in the Symphony Age, that organ system report that we've created, we can say your highest aging organ system is your liver. So we might want to focus on liver health, right? We might want to focus on things that might be driving it. Might even recommend additional biomarkers to measure that process a little bit further. And so now we can actually make recommendations on individual processes. One of the other big things is a really new thing that I'm super excited about, which is called epigenetic biomarker proxies. And so these are how you can actually predict metabolites and proteins and actually the lab values you would get from a physician with just DNA methylation alone. And this, again, gives us a lot of information. And in some cases, we've shown that our DNA methylation prediction of these things are just better than the classical measurement. So the best example is C-reactive protein. Our prediction of C-reactive protein is actually more highly correlated to every disease outcome that CRP is correlated with. It's more precise, and it has, I would say, not just a better effect size, but more significance. And so we think that our CRP is just a new biomarker for the same process you're measuring for CRP.
So it sounds,
we think we can do that a lot.
It sounds like maybe, and correct me if I'm wrong, but it sounds a little bit like trying to measure your blood glucose versus measuring your HbA1c. If you measure C-reactive protein at any given time, it may be varying and fluctuating, but it doesn't really give you the kind of picture of what your overall state of glucose levels have been. HbA1c does that. And now you can say with C-reactive protein, we're going to do the same thing by looking at DNA methylation patterns. We can tell you what your average C-reactive protein level has been over a longer period of time. And it sounds from your nodding, it looks like from your nodding that's the right way to think about it. You said a lot there, and I want to sort of break this down because I think maybe the listeners will appreciate this. The first generation clocks were good, but they were really useful only for confirming whether or not you were lying about your age. When you say, "Hey, I'm 55 years old," those first generation clocks can say, "No, you're 45," or "No, you're 65." And that's great if you're going on a date and you want to confirm that the person you're going with is actually the age that they say they are. But beyond that, it wasn't really helpful from a health standpoint. Maybe from a pathologist standpoint or a crime investigator standpoint. The second generation clocks were valuable and are valuable more from the standpoint of taking a single snapshot and saying, based on the average population, you are seeming like your body is either aging faster than the clock, or hey, you're beating the calendar. You're aging more slowly relative to the general population. And what you're saying now is that the technology has grown to the point not only that you can take an individual and look at that individual and say over a longitudinal period, you are aging more slowly or more quickly than the clock for you personally. But even beyond that, you can now say, and the aging process is affecting you more severely in your kidneys than it is in your heart, more severely in your brain than in your digestive system, or something along those lines. Is that the way? Is that
That's
the right
exactly
way to look at it?
And I think that our conception of aging just needs to get, I think, more accurate and broken down further. And so that's sort of the idea, is that not everyone ages in the same way, right? Smokers are going to have increased lung aging, right? People who are, outside all the time are going to have more UV exposure, more skin aging, right? And so not everyone ages in the same way. And we need more of that resolution to make sure that our efforts are the most efficiently utilized, right? That we're spending the most time and effort in the areas where we need the most help. And you're exactly right about the generations as well, in terms of, it really comes down to just picking out a better signal. And the better our signal is, the more we can read that, the more predictive we get of what that means, right? And then we can intervene in these processes. And again, going back, I also to that C-reactive protein, we actually call it the HbA1c of inflammation. It's exactly what we say. And I always reference, Dr. Horvath himself, who says that another benefit of using DNA methylation biomarkers instead of plasma is that they represent longer averages and are not as affected by day-to-day results. So we can get a good overview of a lot of those things that are happening, especially things like C-reactive protein, which contains 1,000-fold within an hour. We can get a good overview of sort of where you're at, and that can be a lot of times better.
So let me ask a quick question about this, because epigenetics affects protein expression. And when you talk about changes to the epigenome, when you see changes to the DNA methylation patterns, that suggests that there are changes in what proteins are being expressed by cells that may deviate from the ideal. And in fact, that's what aging may be, is if you believe the epigenetic drift theory, that as we age, we lose that control over what proteins a cell should be producing versus what it is. Talk a little bit about the importance of that control and how that relates to diseases. For example, obviously the biggie would be cancer. And is there a way for you to look at a given organ or given tissue and say, hey, based on the methylation patterns, we've seen a drift away from the ideal to a state in which certain proteins are now being expressed because they're not methylated, they're not being locked away anymore, and you're at risk of cancer. Are you basically talking about, and is there the potential for you to be, I almost want to say like a diagnostic for cancer, but even before there's a cancer cell, it's just because of the methylation pattern, you have drifted into the danger zone.
Yeah. So many things there that are worth mentioning. I think the answer ultimately is yes, we do think we can do that. And I'll talk a little bit about why. One of our newest publications is going to be with the Mayo Clinic, where we actually can predict hepatocellular carcinoma from blood-based methylation before it occurs, and we can actually diagnose it at a 95% accuracy. And that's off 200 patients we trained it on, just 200. So these signals are quite clear for some things, but less clear for others. Hepatocellular carcinoma is one of those things that is quite clear, and we were lucky enough to get it. And really, the outside of what we're doing with aging, the biggest commercial application of DNA methylation as a technology is in liquid biopsy testing. So these are the diagnostics of cancer that take place with cell-free DNA, so DNA in the plasma that comes from cancer cells, which are growing and proliferating, and they're therefore lysing. And as they lyse, they release their DNA into the plasma, and they can be found early. So at stage 0 or stage 1, you can detect cancers you wouldn't have been able to detect previously. I think that's important context for just generally the field. But as we go into, I think, going back now to this sort of theory of, can we predict it early by just this dysregulation that happens with aging? This might be a little bit, I would say, too scientifically technical, but I want to mention it because it's a new breakthrough that happened, and I haven't talked about it ever before. So I'd like to at least mention it here.
That's great.
And this goes to this idea that, when Steve Horvath originally created this original clock, everyone thought it was the groundbreaking discovery. Everyone thought that this is amazing, that this dysregulation or whatever is happening, this pattern is so conserved across tissues, and that we can accurately predict. Now, fast forward, a decade later, what we've sort of found is that you can make effective age clocks with just about any information. So whether it's proteomics, whether it's transcriptomics, and as long as you have enough data in that relative area, you can create a clock out of anything that predicts chronological age. And so it brings up this idea, which is what we call stochasticity. So this is what we call random variation with age. It is basically this entropy, right? We talk about in physics, right? Everything is proceeding to entropy. So the universe is expanding, all of these different things. It's the same with these biological processes, which is that they just have random accumulation of dysregulation across time. And actually that random dysregulation can be as accurate at predicting age as actually picking up on any true biological signal. Mm-hmm. And so now I would say it's a lot less impressive what happened back 10 years ago, because he was picking up on a signal. And of course, it was correlated to outcomes better than even regular chronological age. But ultimately, you could do this with just about anything, any type of data set that spans time. So actually, in Nature Aging, about I guess now 4 to 8 weeks ago, we published an article on the stochasticity. And actually, at the same time, they published 2 other articles on the same process. Everyone was trying to answer the same question, which is, are we measuring true biological function or just random variation? And ultimately, out of the papers that were published, the 3 papers, one was from an amazing group in Germany, another group from Harvard, and then we did this with some collaborators at Harvard and the Chinese Academy of Sciences. And we showed that the stochasticity between those first generation clocks and those second generation clocks greatly varied. And so what we're picking up on those newer generation clocks is not just random signal, but actual biological degradation. And that leads us to this idea that there is a signal we can pick up that predicts the phenotype outcomes like cancer, for instance, like disease, like cardiovascular disease or kidney dysfunction. But we just have to have different ways to find it. And we have to have more ways that are actually tied to what we experience from a health function. So that it all goes back down to what is the signal actually predicting and how do we find it? And the better we get at that, the more we're going to be able to do the things that you mentioned. And that's actually where this is going now. Instead of just doing aging, we also now can do disease risk scores. So these are like polygenic risk scores, that everyone might get if they go to a 23andMe or the Ancestry, where you can look at your genes and predict outcomes. But what we've seen is that these methylation risk scores are better in 2 regards. One is that they're much, much more predictive. The second, though, is that they're changeable, unlike those genomic risk scores. So you're not just sort of saddled with a problem you can't fix. You can actually influence change. And so that's really where this technology is going.
I think that's such a huge and important thing for people to understand. I've worked with numerous clients, and everyone asks me, should I get a DNA test? Like I have this 23andMe or something that I've done. And I've always found it to be a huge challenge because these are one-time tests that do not predict things as accurately as I would like. And literally, you just said it. We cannot do anything about it, right? You cannot change your genes. as a human just is. We need to have some predictable changing outcome that we can create. And this is what's awesome about epigenetics, because we can actually change the expression of genes based on methylation patterns. And these methylation patterns are predictable. So I just want to outline to everybody just how massive this finding truly is. Is it the machine learning capabilities that have enabled this to occur, to step back from just random methylation patterns and random occurrences versus actual predictive outcomes?
Yeah, definitely, and it's just processes that again are able to pick out more of those signals, and it's fundamental. Machine learning is definitely one of those things. The original clocks were created with really relatively crude measurements like penalized regression, and now we're getting into things like neural nets and transformers, which are much more able to pick out the right signals. And again, when we validate those algorithms, we see that we're correct. We're getting closer to predicting those disease outcomes. And yeah, I think you're exactly right. I mean, everyone talks about the Human Genome Project, and if you Google the Human Genome Project, the first news articles you'll see are the failure of the Human Genome Project. Right? Everyone talks about how it's going to give us all of this information and change healthcare, and it certainly helped inform. But we didn't have major breakthroughs that I think we anticipated, and I think the reason for that is, again, it's one small piece of the puzzle. Right? It's from your genomics, you then have your epigenetics. Then from your epigenetics, you have your transcriptomics. Those create the proteins and peptides, create then the metabolites and processes, ultimately leading to your outcomes. Right? And that's not even to talk about the microbiome or cell type percentages or all these things that make it very complicated. And so that's the great thing about where epigenetics sits in this sort of dogma of biochemistry, is it's still at the top. It's still one of the first things that happen to our DNA, but it also changes as a result of the changes downstream. So you have this cause and effect thing, which allows it to be really good at showing us a current status.
So let me ask a question about the mechanisms by which this DNA methylation drift occurs. Because presumably what you want is a healthy, young, teenage person has 98% or some percentage of the proper DNA methylation marks on the DNA. Their liver cells aren't making proteins that are appropriate for a lung cell, and their brain cells aren't making proteins that are appropriate only for kidney cells, and that's healthy. But over time, life, toxins, experiences, infections, sedentary lifestyle, things like that, slowly cause those marks to degrade. And you get this drift, and all of a sudden you got kidney proteins being made in the brain. And that can lead to cancers, and that can lead to other degenerative problems. But what is the mechanism by which those marks are being damaged? Because my understanding is, and this is, and I'm happy to have you correct me if I'm wrong, but listening to people like David Sinclair, or talking to or listening to the ideas of Thomas Seyfried, it appears that your mitochondria generate free radicals and reactive oxygen species, and that they are the primary damaging mechanism by which you lose those epigenetic marks, those DNA methylation marks, and that's what's causing this increased risk for cancer and other degenerative diseases that come with aging. Is that correct? And if not, please, by all means, enlighten us.
So I would say that the jury's still out on a definitive answer, and it is probably multifactorial. But you're right. David Sinclair is probably the most notable example of researchers who's trying to answer this question. And so he calls this sort of the epigenetic information loss theory, right? This idea that we lose information over time, that information that's not properly contained leads to bad instructions. Those bad instructions lead to bad outcomes, right? And he has a model to I would say test this, which he's had some success with, where they induce DNA damage breaks. So they're basically increasing, this damage, and then they're seeing, does these epigenetic clocks accelerate, and do the functional outcomes get worse? And the answer is that yes, they do. And so I think that where the jury's still out. I think there's probably multifactorial. I think, when you talk about, mitochondrial dysfunction, certainly one of those big hallmarks of aging. But things like, antioxidants that maybe help that don't always seem to help. There's actually some data that says antioxidants may be worse for the aging process. And so it's, I think we're all sitting around the same hot spots, right? Which are certainly, proteomic dysfunction, nutrient sensing issues, all these things we categorize as hallmarks of aging, but we're not sure how that direct regulation is happening yet. I will say, though, that this is probably the most exciting question, in my opinion, in science at the moment, period, and all of science. And I'll tell you why. And that is because of this new trend of what they call cellular reprogramming. And so I'll try and introduce this topic at least briefly, because this is the secondary aspect of people's work like David's, which is now that we know that this is degrading, how do we reset this? And so in 2012, Dr. Yamanaka won a Nobel Prize for using Yamanaka factors. And the idea here is that you can take any cell in the body and reset it back to a pluripotent stem cell. So a cell with unlimited potential. And the way that this happens is by epigenetically reprogramming. So we take off the methylation marks and we restore it to where a cell can become anything and do anything. And this is just a huge breakthrough, but it's also very, very interesting for a couple reasons. One of the most interesting reasons is, how does the cell know what to reset to? This question of, genetics is a hard-coded thing. That's not changing. The epigenetics is being reset. But how do we know how to reset it properly? How do we know?
Yeah, there's millions and millions of methylation marks that need to be reset. I'm so happy you brought this topic up, because it's one that I actually happen to agree with you completely. Yeah. This is the most interesting question in medicine right now. Yamanaka's work was originally focused on taking cells, somatic cells, and reversing them by causing the expression of different proteins. And the OSKM, 4 proteins that we all express, every cell can do it. And when they're expressed at high enough levels, you get this sort of reverse aging and reverse differentiation of the cell back up into that pluripotent state. But as you're alluding to, the really interesting thing, and to me, it's almost like that the story about the Babel fish from Douglas Adams, which was like sort of a dead giveaway that God exists, is this idea that when you begin using these OSKM factors, when you begin to activate them, the very first thing they do is reset the epigenetic marks to where they were supposed to be, sort of at the very, very beginning. So you basically can take an animal and move it back from being 65, 70 years old, so that at least from an epigenetic perspective, DNA methylation marks is like a 15-year-old again. Exactly. And I'm not just for the aesthetic people out there. Yes, fur changing, less gray in your beard. All of these things appear to be in the realm of possible, and not just Star Trek-y stuff. So I'm with you. I think it's fascinating. So I actually just came back from Barcelona, where they have this Gordon Research Conference on aging. And one of the tenets of the Gordon Research Conference is that you can only share unpublished data. And so it's a great way to see what people are working on. And I was shocked to see, I went to this 2 years ago. I now was back here 2 years later. And the amount of cellular reprogramming data that have been created in those 2 years is absolutely incredible. We're seeing, obviously David Sinclair's work has shown that you can restore age-related vision loss. So blind mice are now regenerating vision. I think they've done it in humans in Japan. I think they've actually done it in humans.
Yeah, and we're seeing this now in almost every tissue. And every tissue is showing very similar things, which is you can have functional rejuvenation. Again, the protocols, which of those 4 factors they're expressing for how long, they're all different, and it's a slightly different cocktail for each. But what we're seeing is that whenever you rejuvenate these tissues, it is a long-lasting rejuvenation, and we know the mechanism is epigenetic, right? And so I think that's fundamentally, that's exciting. The fact that we're now actually turning back the clock, and not just on these molecular biomarkers, but an actual physiological function. Some people might still have skepticism of any of these ways to measure this idea of aging, because even the definition of aging is still relatively complex. But things that people can't argue with are the functional restorations. Things like, are your kidneys functioning better? Are your heart functioning better? Is your brain functioning better? And those things are starting to be proven with these epigenetic reprogramming. And I should mention, all of those epigenetic clocks are reset in the same way with partial reprogramming. So if you partial reprogram for, 2 hours versus 24 hours, you see, again, differential responses, where 2 hours has some of the age rejuvenation, whereas 24 hours might have even more of that. So it is looking like there's a universally connected theme here, and now we're starting to see the impact on function, which makes us feel like we're in the right place.
So I know David Sinclair is working on small molecules that he's hopeful will have a similar effect to the protein, the OSKM proteins. And you're right. I know there's some people who are removing M, I think, from the mix. It's just OSK, depending on the tissue type. But here's a question for you, and I don't know the answer, and I'm wondering whether or not it's unpublished, and maybe you saw it, and maybe you can give us another scoop, which is DNA methylation is one of several epigenetic mechanisms that exist in cells. It's certainly one of the more important ones, but you also have histone modification. Just for everybody out there, histones are a group of proteins that DNA is wound around, because in order to keep that 3 billion base pair string of DNA organized, the cell wraps the DNA around these protein clusters called nucleosomes, and you end up with a very organized chromosomal structure as a result of these coils. When you modify the chemical attachments, much the same way the methylation is an attachment to the DNA itself, you can attach chemicals to these proteins, and that's histone modification. And that's also an epigenetic phenomenon that controls whether or not that DNA will unwind and be expressed. Similarly, there's non-coding sections of RNA that will interfere with the reading of transcribed genes into proteins. So you can block the protein production sort of downstream from the DNA. All of these are considered epigenetic mechanisms. Question is, when you use these rejuvenating factors, the OSKM proteins or the small molecules, do you see a resetting not only of the DNA methylation, but is there a resetting of the histone modifications and of RNA or non-coding RNA like microRNA or piRNA? Are we seeing changes in those expressions back to the levels that they were at earlier in life?
Yeah. So I think that the short answer is yes, we are, particularly for histone modifications, which have been the most looked at. But I want to have some caveats to that question, which is that that although we're seeing it with histones, the reason we see it with DNA methylation first, and why so much work has been done in DNA methylation, is it's just fundamentally an easier measurement to get at scale. Right now, histone measurement is expensive. It can be difficult to get a lot of data out, so you're paying a lot of money for a little. Whereas with DNA methylation, we can get a lot of data at a relatively smaller cost. And so a lot of these questions about, are we seeing these other things reset? We don't have a lot of data on. But there are certainly data sets now that have shown that the reprogramming also affects the histone acetylation. And so as you're mentioning, that those are things that coil your DNA. So it's wrapped really tightly and can fit in your cell. I liken it, the acetylation has a positive charge. So it's like two oppositely charged magnets. If you try and push them together, they repel. That's exactly what's happening with your DNA. It's repelling that protein from another, opening it up. And when it opens up, your actual transcription machinery to make RNA can actually get there. So we're absolutely seeing rejuvenation of histone acetylation marks that are, again, functionally, but also chronologically correlated. And I think that is super cool. I can't speak about any of the other transcriptomics, probably a little bit outside of my area of expertise. But certainly the histone modifications are happening and corresponding with Yamanaka factors.
It's like the Babel fish again. I mean, this is a gift. This is an incredible gift. There was no reason why it had to be this way. There's no reason why that we can discern right now that it had to be that the DNA methylation marks needed to be replaced, the histone modifications needed to be restored prior to the de-differentiation of the cell back up to the pluripotent state. Because once you start seeing that de-differentiation taking place, then that's not something you can use in a living being. You need differentiated cells to function. I liken it to sort of a kindergartner who goes to school and she's precocious and she looks like she could be anything from a heart surgeon to a college professor in journalism to an astronaut. And slowly as she progresses through her education, she will become focused on one or more things, and it will necessarily mean that some of the other things that she had potential to be aren't going to happen. But, she might move down to become a doctor, and then might be a surgeon, and might be an orthopedic surgeon, and might go into spine. And, all of these differentiation steps make the person more capable, more productive, but less potential. As you move that person back up that age, that age cascade, they gain more potential but less capability, because you aren't going to ask a kindergartner to do spine surgery. Same thing with your cells. There was no reason why the first thing that would happen is the methylation marks would be put back in. It's just so fascinating to me that that all of that happens first. It's an incredible opportunity for extending longevity of and lifespan and health span by decades, if not, I'm going to go out there and say hundreds of years. I mean, it literally could be one of those things where people go in and have this 4-day treatment and they come out and they were 65 when they went in, and a year later you look at them and they look like they're 25, and they really are.
Yeah. Definitely. So it brings up, this idea that I've always been a very big skeptic of, this idea of longevity, escape velocity, or is our scientific advancement of age better than our current aging rate, is that idea, so that we'll continue to live longer and longer lifespans. And I've been very, very skeptical of this idea, mainly because aging is really hard to fix. It's a really, really hard process. No one has beaten it yet. No one has even, relatively come close to the theoretical opportunities we have there. So the idea is, I think, are we close? And I think the answer is I'm now thinking we're way closer than I thought. Maybe it's not that we go in and reset all of our bodies, but maybe it's that we start, taking tissue samples from our bodies and growing organs outside of our bodies that we then replace. So we all get, our own tissues grown and replaced at frequent intervals to improve function. And some of these things are already existing in some degrees, like young plasma, where we see that, plasma from younger people rejuvenates older people. And so I think that I'm super exciting time to be in aging. But I also think it's a super exciting time to be in these signals, because they don't just apply to humans as a universal human being. They can apply to, for instance, dogs. There are companies out there now that are testing dogs and giving them rapamycin for 33% increase in lifespan. There are companies out there that are looking at now with climate change, why broccoli with climate change is now epigenetically changing to cauliflower because of the different climates that are expressed. There are a multitude of examples that this is just a fundamental part of biology that we need to understand. And once we understand it, our application and ability to pull on these levers and switches gets so much better.
So let me ask you a question, because you brought up rapamycin, and there's some other drugs that also have similar benefits, things like 17-alpha estradiol, and people have talked about metformin. I'm not quite so much of a believer. It's a wonderful drug, but I'm not certain that it's going to have the same kind of life extending benefits as rapamycin. But tell me, does rapamycin have an effect on DNA methylation patterns?
We are about to publish a study with Yale that we did with a physician group called Ageless RX, where we had very few patient numbers. We had 3 groups, a 6 milligram dose, a 10 milligram dose, and then a control. And we had, I think maximum of 9 people per cohort. So really, really small groups over the course of a year's worth of time. We weren't able to find significant epigenetic changes in any of those groups, but the trial was really low powered. And beyond that, it was using compounded rapamycin, which is usually only available at 33% of the dose. So probably lower doses of rapamycin. I would still say that even though I don't have direct data on an interventional trial, I would certainly predict that rapamycin is one of those things that certainly improves these epigenetic clocks and improves just overall longevity. Recently, so I mentioned the 33% increase in canine lifespan. That study had been done, but also more recently, one in monkeys was done with a 15% increase in lifespan. And lifespan increases are hard to come by. Even, I think, as you mentioned, those ITP trials, things like the 17-alpha estradiol have limited lifespan increases. And even then, they only worked in males. Right. So I think that the things like the rapamycin, anything that inhibits mTOR, like caloric restriction, like rapamycin, I think is a really safe bet to have probably some beneficial effects with aging. And we're doing a couple other trials now on rapamycin.
Does caloric restriction? You're absolutely right. Rapamycin is mTOR is mechanistic target of rapamycin. So my question is there a change in the methylation patterns associated with caloric restriction, and can you get the same benefits with intermittent fasting?
Another good question. The calorie trial is probably the most notable large-scale trial on caloric restriction. They did 2 years at right around a 10% caloric restriction. So, taking all the calories you burn per day through exercise and your basal metabolic rate and multiplying that times 0.9, that's how many calories they were intaking. And what we saw in that trial was physiological improvements, but also reductions of certain biological clocks like that Dunedin pace rate of aging clock that we have as a result of the development of Duke and Columbia. So I think we have some really good data there to suggest that caloric restriction does help reduce these clocks. The question of intermittent fasting or time-restricted feeding is one that we don't have data yet. But at this conference, there was a group led by Chiara Herzog, and she did show that there was some epigenetic age rejuvenation in time-restricted feeding. So only eating within certain hours. It brings up this question about, intermittent fasting or, these multi-day protocols like Valter Longo's, Prolon type diets. We're doing some of those testing too. And so I hope that we'll be able to show that as well. But I think that generally things that, inhibit mTOR, increase autophagy, generally pro-aging, or I should say anti-aging, better for optimal aging, I should say. And so I think that again, a lot of these assumptions being proven in this data is what confirms we're measuring the right thing. Because if we already know it has functional improvements and we're seeing the same change, that makes us feel really good about it.
Because you brought up the concept earlier of different organs being targets of breakdown or methylation in certain areas and patterns driven accordingly. Have you noticed, or has there been anything, and I realize this is getting, we're questioning downstream from where we're at, I think, right now. Is there any difference in terms of mTOR activation in muscle versus, skeletal muscle versus other tissue? Because we do know, and this is that huge debate of mTOR being on in an anabolic state for muscle tissue, but being off in many of the other tissues at certain rates. So have you noticed, or is there anything that you've come across?
Yeah, so we actually did a study with Auburn looking at muscle biopsies of people who were doing exercise or differential weight exercises. And one of the things we looked at was the regulation of mTOR levels. And so, what we found there was what you would expect, which is that in proliferative anabolic phases, as a result of increased muscular activity, we see down regulation. I would say we see upregulation of mTOR. Although we did not compare that to the rest of the body. So I should say that, we are looking at that in the conversion of maybe which tissue is most applicable. But at the moment, we don't have any good data. And I think that is some of the people's fear, even about things like caloric restriction, is that even protein restriction, like methionine restriction, for instance, that if you're doing that type of restriction, are you improving aging or hurting one of the worst outcomes of aging, which is frailty and muscle mass loss and sarcopenia. And so it is, sort of one of the big questions, I would say, and one that I don't know that we have an answer for yet, but certainly one that we're looking at. And I think that methylation could certainly help us solve.
Excellent. Yeah. I'm wondering if there's I've been thinking about this for myself personally. Is there a pulsing, if you will, that is appropriate, like annually? Do you spend like January and February in an anabolic state where you're, trying to grow muscle and build it, but then spend the balance of the year in intermittent fasting and trying to minimize the amount of time you spend with mTOR active? And does it depend on what time of life? When you're young and you're healthy and you want to get stronger and be an athlete, is that the time to spend 11 months of the year in that mTOR dominated anabolic state and only 1 month in the catabolic autophagy mitophagy driven state to just cleanse things, but you're basically good anyway? And that by the time you get to 65 or 70, you want to be spending some time in that anabolic state, but most of the time in that cell preserving, DNA methylation preserving, slow aging, caloric restriction state.
Yeah. And I would say that's actually what most of our physicians are doing now. They're thinking generally about things like 9 months on mTOR inhibition protocols, 3 months off. That way you can, get so I think sort of the best of both worlds. I'm also a big fan of this. We don't have to go into it, but this hyperfunction theory of aging. This idea that, that we grow to sexual maturity, and part of that problem is we don't ever sort of stop. On that pathway, and then dysregulation comes. And that's one of the other data points I really like to always mention is that in several studies, the earlier you start rapamycin, the more of an effect it has. And that I think, that sort of hyperfunction theory of aging is led by Mikhail Blagosklony, who's one of the first and early predominant users of rapamycin in the clinical setting. So again, I think that there are a lot of different theories out here, but I think we're getting closer. We're getting closer to these ideas on aging and really functional definitions and surrogate biomarkers that are actually effective in clinical use. I think that we can talk about all the things we don't know, but I think it does go also to, we know a lot. And already I think these are functionally clinically useful. We found that, you can predict things like, cancer outcomes as a result of biological aging. You can predict response to spine surgery better than traditional frailty measurements. We talk about a lot of what we don't know, but already I think these are functionally useful tools in a variety of areas, and they'll only continue to grow in that area.
So as we sort of come to the end of the hour, is there an opportunity for people, either clinicians or individuals who are interested in getting access to this testing? I mean, I understand it's not something that needs to be prescribed. People can go and do this. This is the opportunity to sort of plug TruDiagnostics and the offerings that they have. I'm a big believer in this. I think this is a wonderful technology. I'm encouraging anybody who has the ability to study this either longitudinally themselves or in cohorts of people. I mean, we should rally people together and put together some sort of like volunteer cohort that wants to do this. What is it that you offer people right now, and what is your best selling products, and how does it all work?
Yeah, so generally what that looks like for the actual end user is we take a blood sample via finger stick blood spot card. So usually you prick your finger, collects a few drops of blood, it comes back to us, and about 2 to 3 weeks later, we generate results. And right now, those results mainly report on aging, telling you things like, again, those organ system ages, things like your overall age with our omic age predictor we created with Harvard. We have our rate of aging, so how fast you're aging right at this moment. That's, again, probably one of our favorite algorithms. It was created by researchers at Duke. And then we have a whole host of other reports, things like we talked about weight loss and caloric restriction. We can predict if you're likely to lose weight if you restrict calories, which, as we talked about frailty, is something good to know, right? Are you expected to lose a lot of weight? We can actually read how much you've been smoking or drinking in your DNA methylation patterns. We can predict your fitness behaviors like your VO2 max, your grip strength, which we obviously know are important functional measures with aging. And that's really, I think, even just the beginning. Right now, that's the information we have on our reports. And soon we're going to be coming out with things to quantify nutrition. We'll be able to tell you your alpha ketoglutarate levels, your spermidine levels, your omega-3 levels, all with that same data. So we can actually just even upgrade those reports. Again, it's only from a point in time you took the test, but it's still something we can continue to add on. And then eventually even methylation risk scores for a multitude of diseases where we can tell you your risk and then maybe even recommend how to mitigate it. So a lot is coming, but right now it's a functional age sort of report, and we really are trying to get people to compete for the largest age reductions, trying to protect themselves as much as possible by getting their biggest risk factor down as far as possible.
And how many different methylation marks are you measuring, and how many different proteins and genes are you looking at? Because I think you quoted some astronomical number. I mean, more data is better, but wow.
Yeah. So we're measuring about 1 million locations in your genome, and basically at each of those locations we get a number that's a percentage of methylation out of all the copies we've tested. How often is it methylated? And so it's a number between 0 and 1. And so we get that about a million, a little bit under 1 million locations. And I think that's also important because we've taken this idea that the data is really important. We don't use it all in our reporting, but for instance, we're able to add symphony age because we measured those things previously, and we can use it to build better models. And so we measure a lot in terms of the proteins we can predict with methylation. We measured about 8,000 peptides and proteins to create predictors of. Not all of them are functionally work, but we can still predict about 800 proteins with relative accuracy. We don't have that on our reporting yet, but we're using that to improve our prediction of different diseases. So, for instance, including things like tau and amyloid predictions into our predictions of Alzheimer's, or including those HbA1c predictors or fasting insulin predictors in our predictions of type 2 diabetes to help again measure more of that specific biology. And so we think it not only is it maybe a useful tool to do maybe some of this personalized nutrition, but also in the future use it to train algorithms that are really comprehensive for what we care about.
I'd love to get kind of a cool little answer from you if there's anything. Has there been any outcomes that you found that were entirely unexpected? Any lifestyle factors, anything that kind of surprised you in learning what you've learned so far about aging in the process?
It's a good question. There have been a multitude of surprises. I think one that I think was fun and surprising was sort of what we found about lithium. So you have a lot of bipolar patients who are taking lithium as a way to mitigate some of the problems with their mental illness. It's a prescribed therapy, and strangely enough, I think in looking at these meta-analyses, we see that people who are taking lithium age slightly better. And then you look at the literature and you find that many people postulate lithium has a direct anti-aging effect. And so some of those things are pretty cool where we were able to find unique associations. I think across the board, there are another couple examples that we found. One of my favorites is that just as we were starting this analysis of being able to predict proteins and metabolites, we were doing a ketamine trial. Looking at depression and ketamine and how well it was able to be treated, and looking at the methylation differences. And, I think that really we expected that trial to hopefully create predictors of who can respond versus who can't. And really everyone responded, which was negative there. But one of the cool things is what we use our prediction of these proteins, and we found there was only one significant metabolite that changed in all of our thousands of model predictions. And it was a metabolite that is known to be associated with liver injury whenever it's with ketamine. So we're starting to see again that we can predict these things with enough resolution that we can find even subclinical biomarkers that are affected in this pathway. So I think that was also one that was really exciting because it backs up the usefulness, I think, of some of these things and confirms with the literature. So there are a lot of examples, but I think Again, there are a multitude of these that I could choose or pick out. I think some of just my last one is probably, we can even see a generational inheritance of epigenetic factors. So we can even pick out, for instance, people whose great grandparents have experienced high degrees of famine, or we see signatures of the Holocaust or the Rwandan genocide, even passed along through the epigenetics. And so there's a limitless amount of applications, I think, here. And all of them are, I think, very cool to understanding, how we're made and how
this is huge, because it's something that JP and I have spoken about in previous episodes. We had a whole epigenetic kind of section that we did. So anybody who wants to listen, head back there. But I think what's really cool here is we're able to now identify these individual characteristics to really create the most personalized approach to healthcare, to longevity, to health optimization that has ever been available to anybody at a very high degree of confidence for each individual. And if nothing else, this is some of the most empowering information that you're ever going to be able to be given, because this is literally your genes, how they have interacted with epigenetic changes and generational challenges that people have experienced in the past in your family, how you are currently dealing with the stressors of your day-to-day life and your lifestyle, which is massive, and how your genes are being transcribed accordingly. This is, I think, such a huge opportunity for people to really be empowered to take back that full control of their health. And it's very, very exciting to have you, Ryan, on the forefront of this with TruDiagnostic. So for anybody who's interested in getting the testing done, interested in learning more about this, go to TruDiagnostic.com, TruDiagnostic.com. And it's, I think this is honestly, we're on the path to revolutionizing absolutely wellness and healthcare across the board.
Quick point. So when you go there, and I think this is what you said, once you put that blood spot on the paper and you send it in and you do the analysis, yes, it's true that right now there are a set number of tests that can be done. And it's pretty comprehensive. And I think the costs are in the hundreds of dollars, not thousands of dollars. And maybe even the low hundreds. I'm not committing to anything for you, but in the low hundreds of dollars. And you get a report back, but you have that information. And in the future, when you find more correlations and the AI develops more confidence around other predictions, you can reference that same material. Now, again, it's a snapshot in time. So you're only going to get what you were at the time that you put that blood spot on the paper. But there's additional analysis that can be done as the technology gets even better. And if you want to, you can longitudinally look at things by doing this annually or doing this on whatever cadence you think is appropriate. In fact, maybe that's a good question. If you're going to try to see how things are changing for you, do you think it's an annual thing that you should be doing? Is it is it every 5 years, or is it, every 2 months? I'm sure, from a financial perspective, you'd love everybody to do it once a month. But,
yeah, and from the data perspective, but at the same time, we like to be pretty evidence-based in our recommendations about how often you should test. And the answer is, unfortunately, it depends on the algorithm. Because each algorithm can be slightly more sensitive to change. More algorithms can be, I would say, more precise and able to measure, I would say, different degrees of change. Generally, we recommend to not to do this more than every 6 months. There are some algorithms we have, like the Do Need and Pace, which we offer as a standalone at a lower price point, because it does change within 4 to 8 week intervals and can give you some good information. But generally, for the majority of these biological clocks, we recommend no more than every 6 months, so maybe twice a year. And we're getting better at that, so maybe we can do it more frequently with more realistic information. But we also don't want to give you bad information. We don't want to give you something that might be confounded by what you ate that morning, or a period of stress or transient change. We really want to make sure it's actionable, and so for that we recommend no more than once every 2 years. But yeah, I think that with that information you get a really good idea of where you're at, and then also figure out what's working for you. And everyone is I would say very different. And going back to that personalized aspect, I think that is really some of the benefit, is you're looking at your gene regulation and really figuring out what's moving the needle.
This is absolutely
That's
phenomenal. It's been an absolute pleasure having you on the show, and we want to thank you so much for your time. This was absolutely wonderful. For anybody who made it to the end of this episode, thank you so much for listening. We hope to help you upgrade your health on an ongoing basis. Have a great day, guys.